Okay, this is the third video for genetics, and now we're looking at another tool that we use when looking into the genetics of somebody or looking for a disorder or just looking to make sure that everything's fine. And these are what we call karyotypes. And so karyotypes are a picture or an image of the chromosomes. Chromosomes. Okay, and so remember, well, I guess or telling you new things, but a female, when we look at the X or the sex chromosomes, they are the last ones on the karyotype down here. It's a little bit harder to see on this male, but this one is over here. There's two of them. So females are XX, and then males are XY. So females have two X chromosomes. The males have an X chromosome and then a shorter Y chromosome. So sometimes they're hard to see, but you can tell because the Y is going to be shorter. Now also notice on this picture that every spot is labeled with a 1, 2, 3, all the way down through 21. Same thing on the males. You can't see it quite as well, but 1, 2, 3, all the way down to 22. So because there's two spots at each of the 22, or two chromosomes at each of the 22 spots, we call those autosomes, and there are 44 of them because 22 times 2 is 44. And these are for like all of your body chromosomes. Okay, anything that's going to go on in your body, in your cells, in your muscles, for proteins to be made, all of those genes can be found on these type of chromosomes. The sex chromosomes are just these last two on both the male and the female. And there's two of those. And that codes for everything from, well, obviously it makes you a boy or a girl, but there are certain diseases that can be carried on those. But so when a baby is made, mom, the only thing she can offer the baby is an X. So it's actually dad that determines the sex of the baby because either the sperm is going to be a Y sperm or an X sperm. So if dad offers an X sperm, well, then they're going to have a baby girl. Mom donates the X egg, and then if dad gives a Y sperm, well, then it's going to be a baby boy. So 44 plus 2 equals total chromosomes at 46. Okay, so sometimes when we look at these pictures, we'll notice an abnormality in here. And so if you look down below, trisomy 21 is known as Down syndrome. And what happens there is that here you notice down at spot 21, there's 3. There's an extra. So this one has 47 chromosomes. They call this trisomy, tri for 3, somy 21. Okay, another thing that can happen is like a misplaced chromosome where it breaks apart differently and it goes and latches onto something else. So everything's good as we go through this karyotype over here on the right until you notice at spot 15 there's like a whole extra extension on there. And what happened is one of the chromosomes from spot 22 went up and glued on to spot 15. Okay, so that's going to cause a genetic disorder as well. So abnormalities generally are going to be a change in number of chromosomes, like you saw the Down syndrome chromosomes, or a change in length. Sometimes you'll see what we call a deletion, where let's say even over here at spot 5, like half of this leg is just missing. It's gone. Sometimes you'll see it where on, let's say, spot 3, okay, we get an extension where it like gets a repeater and it just kind of like misfires and doesn't copy it correctly and just keeps repeating a certain letter in there or a certain sequence, and that too would result in a genetic disorder. So when we talk about chromosomes, where's the picture? Why is that not on here? I have no idea why it's not on here. Aha, here we go. Okay, so this is a pair of homologous, and when we say any prefix homo means same. Okay, so on that karyotype, when you looked at spot 1, they're the same length, they hold the same genes, they're both chromosome 1 at spot 1. At spot 2, they're the same length, they hold the same genes, one came from mom, one came from dad. So they're homologous chromosomes, one comes from the dad, the other one comes from the mom. So we call that the paternal, that's your dad, this one is maternal from the mom. So this one's just kind of generic, but because they really don't look like this or are that spread out or that big, but this would be a particular gene right here. So dad gave a little r. On this chromosome for this gene, mom gave a little r. So if we're talking about a particular gene for running, okay, running fast, running slow, if you have a little r, maybe you're a slow runner. Okay, so they call each of these spots R, P, A, B, and C. Those are called the loci or the locus. Okay, it's a particular location. And so that's where genetics has gone today. If we know that somebody has a genetic disorder or we're looking to see if their children do, we know exactly where those genes are because they have mapped out the entire genome. And we can go in and look at that particular known spot to see if there's a mutation there. So in terms of looking at this other gene for P, okay, this one, they are homozygous dominant for. So this R gene, they would be homozygous recessive. For the P gene, they would be homozygous dominant. Okay, remember dad gives one and mom gives one. So that's where all of our Punnett squares came from. So in terms of, this child, okay, say that he's, we'll go down to the A's, big A, but if he marries, because these are his two, but let's say he marries a female now that's A and she has a little a, okay, now we can look at the possibilities of their children. So still, we're always going to be talking about homozygous dominant, homozygous recessive, or a heterozygous where you get a big and a little.